All right, we're diving right into the world of superfoods today. We're going to tackle a really important question. When you eat something that's supposed to be healthy, how much of that goodness does your body actually get to use?
I mean, you've seen them everywhere, right? Protein cookies, vitamin-infused snacks, even superfood brownies. It sounds like the perfect answer to our snacking prayers. But is it really that simple? Can you just cram a bunch of nutrients into any old treat and call it a day? That's the exact question we're digging into.
Because here's the thing, and this is a big one. There's a huge difference between the nutrients listed on the label and the nutrients your body can actually absorb and put to work. It turns out the whole journey from your mouth to your muscles is, well, it's a lot more complicated than you might think.
To figure this all out, scientists needed a test subject, and they chose a real champion from the superfood world, spirulina.
So what is this stuff anyway? Well, spirulina is a type of blue-green algae, and on paper, it is an absolute nutritional powerhouse. We're talking nearly 60% protein by weight. It's loaded with antioxidants, it's packed with vitamins and minerals like iron and zinc. It's basically the heavyweight champion of nutrition density.
So, the researchers took this incredible ingredient and baked it into 3 very different snacks. First, a soft, airy cake. Second, a dense little peanut ball. And third, a classic, crunchy salt biscuit. The stage was officially set for a nutritional snackdown.
But how do you test something like digestion without a person? Simple. You build a robot stomach. Scientists use this fascinating lab process called in vitro digestion, and it perfectly mimics what happens inside our own bodies when we eat.
This whole simulation happens in 3 key stages. First up is the oral phase, which is pretty much chewing and saliva breaking things down. Next is the gastric phase, where the stomach acid gets involved. And finally, you have the intestinal phase, which is where our bodies are supposed to do the heavy lifting of absorbing all the good stuff.
And this diagram here kind of lays out the whole journey. It starts with preparing the sample, that's the chewing. Then it moves through those oral, gastric, and intestinal phases, using beakers with different enzymes and acids to act just like our digestive system. But that final filtration step, that's the really important one. It separates the filtrate, that's all the nutrients ready for absorption, from all the undigested bits.
So the experiment is running. The robot stomach is doing its thing. How did our 3 spirulina snacks actually hold up? You know what? The results were pretty surprising. First, let's just look at the raw numbers. Just by adding that bit of spirulina, the protein content of every single snack jumped up by about 2 to 3 grams. The salt biscuit started out with the most protein and ended with the most, almost 19 grams. So far, so good. Everything looks great. But this is just the starting line.
And this is where things get really fascinating. The study showed that what a food is made of is only half the battle. The other half is all about the food's physical structure, what scientists call the food matrix.
Think about it. Over here, you've got the cake. It's soft, it's airy, it's full of little pockets. You'd think that would make it super easy for your digestive enzymes to get in there and do their job. And then on the other side, the peanut ball and the biscuit, they're much denser, way more compact, so logically they should be harder to break down, right?
Okay, this chart right here is the crucial piece of evidence. It tracks how much protein was available at each stage of the digestion process. That black bar, that's the total protein at the very start. But the pink bar, the one labeled intestine supernatant, that's the one we really care about. In the lab, supernatant is just the liquid that has all the good stuff that's been broken down and is ready for the body to absorb.
And this is the big twist. Just look at the cake. It started with almost 11 grams of protein, but in the end, only about 7 of those grams were actually available for absorption. Now look at the biscuit. Yeah, it started with more protein, but it released a whopping 14.5 grams for the body to use. That dense structure, counterintuitively, actually led to a much more effective release of nutrients.
So what's going on here? Well, this quote from the study explains it. A porous structure, like the one in the cake, does let enzymes get to work really fast. But in this case, it might have worked too well, too early. Think of it like a nutritional flash in the pan. The biscuit's denser structure led to a slower, more sustained breakdown, releasing its protein at just the right time and in just the right place, the intestine, for optimal absorption. And all of this leads us to the single most important concept in this entire explainer: bioavailability. It's the metric that truly matters. Simply put, bioavailability measures how much of a nutrient your body can actually pull out of a food and use. It's not about what's on the nutrition label, it's about what actually ends up in your system.
Here's a great way to think about it. The total protein listed on a package is like the total amount of money in your bank account. But bioavailability, That's the cash you can actually get from the ATM. A $1 billion bank account is pretty useless if you can only withdraw $10, right?
So, after all the chewing, churning, and filtering, which snack allowed us to withdraw the most nutritional cash? The winner, for protein bioavailability, clocked in at an impressive 60.1%.
And here are the final results. The salt biscuit takes the crown, making over 60% of its protein available to the body. The peanut ball came in as a strong second. But the cake? Only 23%. That means for every 10 grams of protein in that spirulina cake, your body could only really use a little over 2 grams. The biscuit was more than twice as effective.
So the bottom line is this: we can't just look at a list of ingredients. The way those ingredients are put together, that food matrix, has a massive impact on the nutrition we actually get. The vehicle delivering the nutrients is just as important as the nutrients themselves. And that leaves us with a fascinating question for the future. If we now know that structure is so critical, how can we design foods not just to contain more nutrients, but to deliver them more effectively? This research changes how we should think about everything from protein bars to meal replacement shakes, forcing us to ask not just what's in it, but what can my body actually get out of it?